Hello, thanks for giving me the opportunity to talk here today. I'm Carl, I'm a PhD fellow of the MIMO group at NMBU in Norway. Today I will present a state-of-the-art multi-omic, holo-omic, high-resolution Rumen-based project that we've undertaken and are currently investigating and analyzing the data for. So the cattle is a great example of a holobiont. The cattle hosts a microbiome in its rumen, which is the first of the stomachs that the feed enters when grazing. There is an axis from the digester through the rumen wall to the liver. In the rumen, plant fibers are degraded by microbes into short-chain fatty acids or volatile fatty acids and more complex metabolites that are taken up through the epithelium of the rumen wall. The rumen wall is able to directly transport some volatile fatty acids directly into the blood, whereas more complex metabolites like acetate and propionate are transported through the portal vein to the liver, where they are processed before they become available for the host. So this is why our project becomes relevant. We sampled 71 cattle bulls, 2 breeds, Lewing and Aberdeen Angus Limousine cross. 3 sample locations to capture the holo-omic axis, 3 columns in the table, and comprehensive sequencing of several molecular layers to capture the metabolism and signaling of the biological pathways, and those are the several rows in the table. So for DNA, we have 16S for overview, Illumina for depth, and Oxford Nanopore Technologies R10.4 for length. For RNA, proteomics, and untargeted metabolomics, we have analyzed all 3 sample locations. Regarding volatile fatty acids, we have measurements for 6 common targets in the digester to track the breakdown of plant fiber polymers. And for phenotypic information, among other key performance indices, we have methane production using a respiration chamber method where we see 30% variation between the bulls. So, I want to show you something. For 16S ribosomal RNA sequencing, we had 71 samples, so these are 71 dots, 1 for each bull. When looking at the 16S profile of our samples through the lens of ordination, in this case PCOA, with robust Aitchison distances, the split appears. When we saw this, we freaked out, so we went in to double check that we hadn't somehow biased our samples. We started looking into obvious effects. Methane on the left, evenly spread on both sides. Breed on the right, evenly spread as well. We didn't want to believe that this was a biological effect because there are so many ways that it could be confounded by something else, or even just be a batch effect. We had a lot of batch variables, which I'm showing the data for here. Pen, how the bulls were housed, residual feed intake, how much they ate, the slaughter group, in which batch they were slaughtered, even in which bags the samples were stored in our freezer and how they were delivered. No explanation of the split. Critically, we also tried summarizing the abundances on the genus or family level. There still is some indication of the clusters on the genus level, but it disappears on the family level. A possible biological factor, depending on the taxonomic finer details. What I showed up until now was 16S. So what about the max of the microbes? Guess what? We still see the split clearly. And stepping further into our multi-omics dataset, digester metatranscriptomics, rumen wall epithelium metatranscriptomics, digester metaproteomics, and rumen wall metaproteomics, we see strong signs of the split, at least in the metatranscriptomics and metaproteomics of the digester. In metatranscriptomics, top left, the clustering seems clearly present on both MAC and ciliate data. For metaproteomics, bottom left, there is a bit more overlap between the clusters. Transcriptomics and proteomics are such different methods, where proteomics has redundancy issues, transcriptomics is more specific. So the fact that we are seeing the same split across these different methods is an indication that something biological might be going on. We also had 6 VFA targets, volatile fatty acids, as part of our metabolomics. We see propionic, butyric, isovaleric, and valeric acids being significantly different between the split groups in the digester of the bulls. Specifically, butyric and propionic acids have a large difference. VFAs are important in the energy transfer from feed to host through the microbiome. So this is another hint that something biological is lurking in the split. So we know that this is not a batch effect. So what is it then? This is a MAC abundance PCOA biplot showing samples, bulls, as well as those MACs that differ significantly between the 2 clusters, trimmed to genera with more than 5 significant MACs with taxonomic details provided. What is interesting is that we see that the inferential MACs driving the split seem to often represent different strains of the same genus. There are several Methanobrevibacter and Sodaliphyllus MACs on both sides of the second axis. We think maybe the ciliates are taking part in driving this as well. Here, we saw in the digester metatranscriptomics and metaproteomics that the ciliates follow the split. Here are the 5 ciliate species that differ the most between the 2 clusters. All are increased in group 2. These ciliate genomes are booming with fiber-degrading cazymes. They play an important role in fermentation and butyrate production. So using several different methods is a good way to validate one's results. So we looked into network analysis to see if we could observe and further characterize the split. This problem that we're facing is reminiscent of the needle in the haystack problem. We have an absolute massive haystack of information, but we need to find the signal that drives the split. So we used WGCNA, Weighted Gene Co-expression Network Analysis, which is a framework that does hierarchical clustering to group the proteins into clusters. For each of these clusters, principal component analysis roughly is applied to define so-called eigenproteins, which are idealized proteins that capture the main variation within each of these clusters. To these clusters, or eigenproteins hereof, we can correlate our KPIs and metadata. What you see here are all digester eigenproteins correlated to the split variable. If we then take the top 5 most correlated proteins in these modules and summarize which species they come from, we get something that looks like this. The clades in the panes are phylum and class, and the names next to the dots are genus and species. What we see is that there is a great overlap of these results with the metagenomic split results. Let me annotate which ones are overlapping. So these are the bacteria that overlap with the MAC abundance PCOA of the metagenomes. And additionally, these are the ciliates that I've already glossed over. Isotricha and Ensodinium in group 1 and Epidinium in group 2, which is exactly what I've already seen. Again, this is a completely different approach, but we find many of the same species as with the MAC abundance PCOA, at least for the bacteria. So, finally, last slide. This is the take-home, 2 clusters and good reason to believe that it is biological. What I hope to have shown is, in this short talk, is that there is a need to find the gold standard for integrating the multi-omic layers of holo-omic studies. What we're looking into now is to develop a fully parameterized holo-omic bioinformatic workflow based on correlation of multidimensional scaling and network analysis. Because our dataset shows a novel interaction, we hope, in multiple omic layers, this dataset can be a good empirical starting point to tweak the necessary parameters to build up a workflow that captures relevant interactions within the holobiont. So one of the major challenges that we've met this far is that databases are often limited and too species-specific to capture the variation of complex holobionts. I think we also need to focus on species-agnostic database integration with a robust annotation of metabolic function. Finally, I want to give a special shout out to my colleague Velma Aho, who I worked with to characterize the variation. She will present this more in depth at ISME in South Africa in 2 months from now. Thank you.